start our review for the day. So if you would, please join me for this little review. And remember, the quiz that I'm giving you guys today is 20 questions. It's a little bit of everything from DNA to RNA. There are no questions about mutations on today's quiz. I'm going to save that for a review on Monday before we take our exam. Now, your exam on Monday will include these exact 20 questions. So, you know, this will be a really good preview of things to come. Oh. So we got 27 students in the Zoom right now. So I'll wait till we see 27 people signed up to participate. And I was just kind of looking at our calendar and kind of planning out the rest of the semester. We only have 21 days left of class since the last week before Christmas break is technically an exam week, which for this class, we do have a biology EOC. And since the second period, we'll be taking ours December 15th in the morning. If you're having any technical issues and you need to play along with the chat, please message me right now. Say, I have to use the chat to play the Kahoot if you're at home and having some technical difficulties. Yes. Gotcha. All right, let's go ahead and rock and roll. All of the following are true in regards to DNA, except for what? So which of these is not true? Which of these is a false statement? Excellent work. Yeah, so DNA is found in all living things. So to make a claim such as the one here where DNA would only be found in plants is quite absurd. All living things do contain DNA, even some things that we don't consider alive, like some viruses. DNA is typically found in the nucleus of eukaryotes or in the nucleoid region of prokaryotes, and DNA contains our genetic code or the universal genetic code. DNA stands for what? So what does this acronym stand for? I don't see you in the waiting list. All right, hold on. Let me try to invite you really quick. Who? No, I don't have the mini period, I don't think. Okay. Hold on one second, y'all. We're having some issues here. Why isn't this working? All right, second period. Thank you guys for your patience. Okay, try that. Let's see if that lets you in. I just sent it to you in the remind. Yes, and so the answer is deoxyribonucleic acid. The deoxyribo part is referring to the special sugar found in DNA nucleotides known as deoxyribose, which is simply a deoxygenated ribose sugar. And then DNA is a nucleic acid. It tells us what kind of biomolecule it is, and it also tells us that if it's a nucleic acid, that it's a polymer made of many nucleotides. All right. And we're off. All right, nucleotides are made up of the following. So which of these describe the structure of a nucleotide, which would be the building block of larger nucleic acid polymers? I couldn't tell you last time I had Skittles, but all right. So yes, nucleotides are going to be made of a sugar, nitrogen base, and a phosphate group. You know, if we're talking about DNA, that would be deoxyribose, and the nitrogen bases would be things like adenine, thymine, guanine, and cytosine. And then it really doesn't matter what kind of nucleotide we're talking about, whether it's DNA or RNA, they're all going to have the same phosphate group, right? So the major variations between DNA and RNA actually exist in the nucleotides. And that would be the sugars that are different, deoxyribose versus ribose, and the nitrogen bases, whereas DNA has adenine, thymine, guanine, and cytosine. RNA does not have thymine. Instead, it has uracil. In DNA, adenine pairs with what? Who does adenine like to dance with? And then be thinking why. And you can't just say just because. Yes, that would be thymine. Now, can anyone tell me why adenine likes to dance with thymine? The shapes, yes, right? So an easy way to remember this is simply apples in the tree, right? A with T, or straight line with straight line. You use straight lines to make A, use straight lines to use to unify T or to draw T. But if you really want to know why, it's because adenine is a purine, so it has two rings, and thymine is a pyrimidine, which makes for a single ring. And so that gives us three rings across the ladder rung of our DNA molecule. But that still doesn't necessarily justify why, because why does adenine not pair with cytosine, which is another pyrimidine? And it's because of the chemical bonding sites. Adenine and thymine both have two places where they can form chemical bonds. So they're a perfect match. They form two hydrogen bonds between them. What about guanine? Who's guanine like to dance with? Who does guanine like to pair with? Would they be trying to think about why? Yes, that would be cytosine. And why that is, because guanine is a purine, cytosine is a pyrimidine, and they both have three chemical bonding sites. So cytosine and guanine typically form three hydrogen bonds between them. In fact, there are places in our DNA that are exceptionally sticky. They like to stick together. And you would find a high concentrations of cytosine and guanines there. Whereas there are places where you find lots of adenines and thymines. And since they only have two hydrogen bonds, they tend to dissociate or unanneal rather easily. Purines are double ring structures, true or false? And that is true. Purines have 2 ring structures, whereas pyrimidines only have a single. How can you keep them sorted out in your mind? I always just think you need to see the pyramids before you die. So that reminds me that uracil, cytosine, and thymine are all pyrimidines. It's really quite interesting is we've actually found things like amino acids and nucleotides on various space debris. Right? So they're not necessarily purely of biological origin. They do form in abiotic conditions. Purines always pair with what? This is important because this keeps the width of the DNA molecule consistent throughout. Yeah, it would be the pyrimidines. In 1953, blank and blank discovered the structure of DNA, and I used a rather vulgar term to describe one of these individuals because they did not give Rosalind Franklin credit for her contributions. And unfortunately, by the time everyone found out, she had died of cancer because of her work with X-rays. What is another name for a jerk? And it rhymes with this individual's last name. And that would be Watson and Crick. Now, you're going to see that on my quiz. You're going to see that on my exam. Are you going to see that on the OC? Probably not. Why do I bring it up? Because when I read about it, when I think about it, it pisses me off. Think about how much further ahead we would be. Light years ahead, in my opinion, had we just gotten over this ancient archaic idea that there truly is a difference in mental capacity between a male and a female. Think about how many wonderful discoveries sat on a desk simply because they were discovered by a female. I'm pretty sure we'd still be twiddling our thumbs contemplating the structure of DNA 70 years later had it not been for the contribution of females. When I was really young, and I mean really young, I learned something that's always stuck with me. Two heads are better than one when trying to figure out a problem. So if 50% of your population is female, why would you leave them out? Are you afraid that they might make you feel inadequate? All right, we'll rise to the occasion. All right, if you had to describe the shape of DNA, you might say it is what? How might I describe this structure? Relating both to the number and the overall shape. And that would be a double helix, right? And so this diagram, although it looks very cartoonish, it can kind of tell us a lot about DNA in the fact that, we've got 2 strands here, and these strands would be the sugar phosphate backbones. These ladder rungs right here would be our nitrogen bases, right, or complementary base pairs. And why do you think in this structure, one strand is blue and one strand is red? What do we know about the process of DNA replication? It's semi-conservative, exactly, right? And so this might be indicating to us that maybe this blue strand was present in the original molecule, whereas the red strand is the newly synthesized complementary strand. So semi-conservative simply means that every DNA molecule contains one new strand and one from the parent or original strand. The rungs or nitrogen bases of the ladder in DNA are held together by what kind of chemical bonds? What holds the nitrogen bases together? What holds the complementary bases? What holds adenine to thymine and guanine to cytosine? These particular bonds are not very strong, which is important for the way in which that DNA is both replicated and transcribed. We need to be able to pop it open relatively easily and put it back together. And so those would be the hydrogen bonds. Where do we find covalent bonds in the DNA molecule? These are the strong bonds. These would be the sugar phosphate chemical bonds that hold those strands together. Because I want to be able to pull the 2 strands apart, but do I want the strand itself to fall apart? No, I want that to stay together. So the sugar phosphates are held together covalent. And the nitrogen bases, the strands themselves, the two strands together, their connection is hydrogen bonds. DNA replication occurs blank, cell division. So when does DNA replication occur? Well, it happens during the S phase of interphase. And if you remember our order of operations, not mathematics, but biology, we remember that interphase happens when? Yes, excellent. That'll be before. I don't care what kind of cell division this little cell is going to experience, whether it's binary fission, mitosis, or meiosis. We must make sure that we've got plenty of DNA to work with. So DNA replication must occur before cell division. If not, we're going to have some serious problems because the resulting cells are going to have not enough DNA. During replication, DNA polymerase reads the parent molecule from what direction? Sometimes we call the parent molecule the template strand. So DNA polymerase would attach and it would read in this direction while synthesizing the new strand in the opposite direction. This is a pretty tough question. How do you read a number line? Yes, that would be in the 3 to 5 prime direction. So DNA polymerase attaches wherever the primase laid a primer, and it reads in the 3 to 5 prime direction, just like we read a number line: 1, 2, 3, 4, 5. It's reading from 3 to 5. So what does that mean about the synthesis of our new molecule? The new molecule is being synthesized in the 5 to 3 prime direction. You might still be thinking, what in the world, Mr. Collins? Let me get my little annotation thing right here. Okay, so let's say we've got a strand of DNA right here, okay? And let's say this is the 3 prime end, and this is the 5 prime end. What does that really mean? Well, if you remember, the 5 prime end would just be the strand of DNA that had a little phosphate sticking out, right? So the last little piece sticking out here would be a phosphate. When DNA polymerase reads a template, when it reads a template, it reads from the 3 to 5 prime direction. So it would be reading like, you know, A, T, C, G, whatever, so on and so forth, right? And so it would read in that direction. And what it would do is synthesize the new strand. So who pairs with A? That would be T, A, G, C, right? And it would continue on in this direction. It would continue on reading right here, starting at 3 and reading in that direction. But since DNA is anti-parallel, what do we know about the new strand? What do we know about the complementary strand? It's kind of backwards, right? It's flip-flopped. So this would be the 5 prime end, and this would be the 3 prime end, because it's anti-parallel. So we're reading the template 3 to 5, and we're synthesizing the new strand in the 5 to 3 prime direction. This took me a while to wrap my head around, and I just think, how do we read a number line? 1, 2, 3, 4, 5. That's how it's read, which means it's going to be synthesized in the opposite direction because DNA is kind of an upside down molecule. Okay. Go away. All right, here we go. Which base is found in DNA but not in RNA? So which of these nitrogenous bases is found in DNA but not RNA? If you discovered this nitrogen base in your RNA molecule, you'd think, uh-oh, something's not good. That would be thymine. Now, here is the honors level biology question. If your DNA molecule was successfully transcribed into a molecule of messenger RNA and a T was present, why would that be problematic? Why would it not be good for a thymine to exist in an mRNA molecule? What is the end goal? What are we going to use mRNA for? For protein synthesis, right? mRNA gets translated into amino acids. How did we do that the other day? We had to look at the chart, right? Are you going to find a T anywhere on that chart? No. So would the ribosome be able to fix an amino acid to that particular codon? No, there would be no anticodon, right? No appropriate amino acid to go there, right? And so you would have kind of like a gap in your protein, and that would not be good. What are the three types of RNA we've talked so far about? One acts as the messenger. The other acts as the molecule that transfers and transports amino acid to the other one that acts like the ribosome. It doesn't act like the ribosome. It actually is one of the main constituents of the ribosome. That would be mRNA, tRNA, and rRNA. mRNA, the messenger RNA, is produced through transcription in the nucleus, and this contains a rough draft of the instructions on how to make a protein. And the ribosome reads the messenger RNA and works with the tRNA molecule to deliver the appropriate amino acid and put them in the correct order. What do amino acids make up? What do our bodies use amino acids to form and to make? In fact, if you're an athlete and you take your sport seriously, you might be at GNC getting some of these. Those would be the amino acids because they're used to make proteins. And believe it or not, whey protein actually contains a lot of the essential amino acids for muscle synthesis, which is why it's commonly the primary ingredient in most athletic protein supplements. What is the name given to a group of 3 nucleotides in RNA? So when we read RNA, when we look at messenger RNA, we look at it 3 letters at a time. What do we call that? We're looking at messenger RNA. For everybody? Or just a couple of you guys? Anyone else have a frozen screen? Just a couple, yeah, a couple of you guys. Well, we'll just, the answers or the choices were triplets, RNA, codons, and anticodons. Now, codons is the best answer. Could I entertain anticodon? Yeah, right, because the anticodon is found on the tRNA molecule, and that is important because that makes sure the transfer RNA molecule drops off the correct amino acid. But typically when we're talking about RNA, we're talking about reading the messenger RNA, we're talking about codons. So will you ever use an anticodon for translation? No. Those charts that I give you are for the mRNA codons. You read them 3 letters at a time. What is the process called where mRNA is created or made? So if your computer is lagging, just think, what makes messenger RNA? What process is that? We're taking DNA and we're making RNA. And to remember the order, I always think we must write something down before we translate it. So what's the Latin root to write? It would be scribere. If you're a scribe, you're a professional writer, or you know that little person who sits in the courtroom who's just happily typing away while everyone's screaming and shouting and chatting at each other? That person's a professional scribe. And if you wanted to review that, you would request the transcript, right? The kind of written form. Transcription occurs in the blank of the eukaryotic cell. So where is DNA transcribed into messenger RNA? If we're talking about a eukaryote. That would be the nucleus. What is the process called where mRNA is made in amino acids or made into amino acids, right? So what process uses mRNA to link together amino acids? So what process is mRNA used in? That would be translation. We translate RNA into the language of proteins, which would be the order and sequence of amino acids. The start codon in mRNA. I just threw this in here for fun. I'll never quiz you or test you on this particularly, but I've just seen if you remembered from looking at the codon charts. I also want to include this because I don't want you to be fooled under this idea that every time you do translation, it'll always be this codon, because it won't be. And that would be AUG. Does anyone remember what amino acid that codes for? Yeah, methionine, right? Yep. And everyone always just remembers, they're like, yeah, codes for meth, right? It codes for methionine. Now, in real genes, yes, that would be the first codon you would see, the starting codon. However, in our problems, in our quizzes, and our exams, and on the OC, don't fall victim to that. They will try to trick you. They'll put one of the answer choices with methionine to begin with, and you'll think, oh, that's got to be it. That's starting codon. But for practice problems and kind of testing your ability to translate and use that chart, that is not the case at all. Translation occurs in the blank of the cell. Where does translation occur? Where do we actually take mRNA and finally translate it into amino acids, the language of proteins, completing protein synthesis? And these little guys could be floating around in the cytoplasm, or they could be embedded along the rough endoplasmic reticulum. There's slight variation between these, prokaryotes and eukaryotes. That'd be the ribosome. During DNA replication, blank breaks the hydrogen bond and unzips DNA. So who unzips DNA during DNA replication? It would unzip the helix. That would be DNA helicase, right? Unzips the DNA molecule by breaking the hydrogen bonds. Once the template strands are exposed, well, remember, topoisomerase would be holding everything still. Primase would attach and lay down a primer, which would tell DNA polymerase where to start. DNA polymerase would read from 3 to 5, synthesizing our new strand in the 5 to 3' direction. But unfortunately, because DNA's anti-parallel nature, one side of the DNA strand is replicated in chunks, and we call those Okazaki fragments. So they need to be later glued back together by ligase. DNA is double-stranded and RNA is single-stranded. True or false? Some of you guys need coffee, exercise, and a good night's sleep. Some of you guys look like the Walking Dead right now. That will be true. DNA is double-stranded, containing 2 sugar phosphate backbones wrapped around each other in the double helix, whereas RNA tends to exist as a single strand. Now, believe it or not, RNA can actually kind of fold back over on itself when you eventually take molecular biology or biochemistry or even a higher level biology. You'll actually start to look at the structure of tRNA. It's kind of wild. It's a single strand that kind of bows back over on itself, which is what exposes the anticodon and allows it to carry the amino acid. The DNA, is it smaller or larger than RNA? Is DNA smaller than RNA? What do you think? You had to take a wild guess. That's false. Because think about it. One gene found on one chromosome could code for a single RNA molecule. And one molecule of DNA can contain hundreds, if not thousands of genes, right? And so a single DNA chromosome could be 1,000 times larger than the biggest RNA molecule. I'm never going to throw you a quiz question or exam question like that. Just DNA and RNA contain identical sugar molecules. True or false? Do they contain identical sugar molecules? And when I mean identical, I mean identical. Because in chemistry, slight variations can have very profound effects. And that would be false. One has deoxyribose, that would be DNA, and the other has ribose. And it's literally just one oxygen missing that creates the difference, but it has a very profound effect on the nature of the molecules. There are many medications out there that actually have two different structural conformations. One is medicine and the other one will kill you. So biochemists just have to routinely check to make sure that the medicines are pure. Transcribe the following DNA strands. So there's some DNA. Give me messenger RNA. Give me the complementary messenger RNA strand. Who pairs with T, A, who pairs with A. You want to say T, but you have to remember that we're talking about your RNA, so it's going to be U. So it'd be A, U, G, 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 G. Okay. Now, when I was in high school in the oh so beautiful Mrs. Soane's biology class, and we were learning about this, I really enjoyed the whole apples in the trees, cars in the garage. Everything just, it made sense. It was easy to remember. Straight line was straight line, curvy was curvy. And then she starts talking about RNA, and I just think, oh my gosh, you got to be kidding me, right? Why are you going to throw me that curveball? So I remember sitting there in class, and this is what I drew. And don't laugh at my drawing. It's a donkey. And what's another name for a donkey? An ass, right? And so I wrote on the hind end of the donkey, you are an ass. And I've never forgotten from that moment that if we're talking about RNA, I'll never forget that, oh yeah, uracil. All right? Guys, remembering things is your responsibility. Every single person's brain in this room has a different way of assimilating information. I like cartoons. I like to laugh. I like to try to be creative, right? And yes, you could just simply pound it into your head over and over and over. RNA, uracil, RNA, uracil. But is that really going to work? Right? You come up with a little story like this. Like, I'm not going to forget this, right? Granted, I teach this for a career, but I had no problem remembering this when I was y'all's age, right? No problem. In fact, I got to college biology and I was like, by golly, I remember one thing, and that was it. Okay, no, I remembered other things, but and I'm not using profanity. That literally is another word used to describe donkeys because they're kind of stubborn. I don't know if you've ever worked with a donkey. right? They can be the most fun animal or the worst. They can be an absolute nightmare. Okay. Translate the following mRNA strand. Now, don't be scared. This is different than what you've seen, but I'm testing your critical thinking. I guarantee you can figure out how to use this. I know it's tiny. I'm sorry. So I've given you mRNA. All I need for you to do is translate it. Uh-oh, we're running out of time. All right, let's take a look. How do we use this? Okay, so first one is CCA. So you find the first space, C, C, A. So our first amino acid coded for by the first codon is proline. The next one is AAG, A, G, the lysine. And the last one, UGA, UGA. So stop. And there is our match. So a couple of my good friends in college were pre-law students. And I'll never forget one time my friend Jay Lutz was studying for the LSAT, which is kind of like the lawyer version of the MCAT. It's like you're not going to law school if you don't do well in the LSAT. And the LSAT's a really difficult exam to study for because it's a logic exam. You're not just studying like what law is this or what happened in this year in America, right? And one of the questions on his LSAT practice exam was one of these. And you might be thinking, like, what does that have to do with law? I could give this, I guarantee you, to some middle school kids and some elementary school kids, and they could decode it, right? And what do we use to decode things? Logic and reasoning. Some of you guys, when you first saw it, you thought, oh crap. But almost instantly you said, okay, it makes sense. The biggest letter is the first letter. The next biggest one is the middle, and the smallest one will be the last one, right? And so you take high school biology to learn about biology because, well, it's kind of like what's going on in your body 101, but also to increase your critical thinking skills, right? Your ability to be presented with a situation and to be able to find patterns and derive meanings. And I don't care what career you go into, this is a part of life. And some of you all need to use this more in like your day-to-day interactions with other human beings, right? Because some of you guys are like, you know, all women are crazy or all men are pigs. And you're looking back at all your relationships and what's the only common denominator? You. Right? So think about that for a second. Look for the patterns, right? You've had 500 boyfriends or 500 girlfriends and you think they're all the same, but the only thing that was the same throughout all of that was you. So maybe there's something that you're not doing right. Simply just not choosing wisely. All right, so let me write down our victors. Excellent work, y'all. All right, so that's all I have in regards to the review. If you have any questions, you can send me a reminder, an email. You guys know what you need to get done. The video questions for 6.1, 6.2, if you haven't already, you've got the interactive, you have the study guide that's due on Monday, and then you have your quiz that's due at 11:59 p.m. tonight. Well, I hope you guys are Godspeed on your quiz, and I hope you have a wonderful weekend. Go spend some time outside, and I'll see most of you guys on Monday.